Good morning to you. Thanks to the organizers. And thanks to you, I have this opportunity to present my work here. How do we see and why do we breathe? This is the question. You would imagine this is the 21st century and we would have the answer. I say no. I say no. Whatever you read in textbooks, they are not right. Why do we breathe? Oxygen, why do we take it in? And how do we see? The process of converting an image in the retina, how do we do that? So I'll try to answer this question with a concept, a new concept called Murburn concept. I have termed this. And this is a paradigm shift idea in biology and medicine. Okay? So before I move on, I would give you a brief idea about Murburn concept, which I'll come back to later. Please, next, thank you. So Murburn concept, many of you would know what the concept of enzymology, Fisher's lock and key, Koshland's induced fit, you know, enzyme substrate binding. It's a very small twist to that concept. It's a very, very small twist, but it's a very, very important one, nevertheless. You have the enzyme. Do you have a stick or something? I can use a stick. If you have a stick, it'd be nice. So basically, you have the enzyme here. And then you have a diffusible reactive species. What the contemporary mechanism of biochemistry details is enzyme identifies the substrate at its active site, and you have a selectivity and specificity because of that, right? What I'm introducing into this picture is that some enzymes can use a diffusible reactive species to catalyze reactions, group transfers like phosphate, hydroxyl, chloride, chlorine atom, all that, and electron transfers in biology. So what you have is enzyme, diffusible reactive species is either produced or stabilized on the enzyme, and then the diffusible reactive species come out. I need a stick. If you can give me something, I will not touch the board, but otherwise I need a pointer. Can you give me one so that your audience can follow me? One simple stick would do. I should have asked for it. Okay, so otherwise I'll have to come like this and show, which will obstruct the vision of other people around. All right. Yeah, that's good. That's good enough. So yeah, that serves. So now, so the Mured burning is the concept from which it is abstracted. Mured means closed. Burning is an oxidation process. Very simple concept. So since the reactive species is outside, there is a zone in which it can interact with ions and generate radicals. So this is a mechanistic outlook. So before we move on any further, go. I can take care of it. Please sit. Yeah. So the layout of the talk is in three. First, I'll talk about vision, the molecular physiology of vision, the classical explanations, and then the Murburn explanation, the molecular mechanism of respiration, the classical one, and my theory, and then the details of Murburn concept. So before I start out, I want to pay my tribute to my mentor, my godfather, my true scientist that I have seen in my life. Lowell Hager was a professor of biochemistry at University of Illinois, Urbana-Champaign. He passed away in 2014. I used to work with him in 1999-2000. And there I was working with an enzyme called chloroperoxidase. It's a heme enzyme. He discovered the enzyme. And I had serious differences in mechanistic perspectives with my host there. So, and there I challenged so many aspects of classical mechanistic enzymology. The perspective was reactive oxygen species is critically inhibiting the enzyme. I showed that it doesn't. Basically, the peroxide depletion is what leads to inactivation. It's not the peroxide-mediated deactivation of enzyme. So basically here, I challenged certain classical perceptions, and he allowed me to challenge it. That's very important. You need to have people who are magnanimous and gracious enough to accommodate that difference of opinion. So I was working in his lab and I challenged his ideas. So the journey starts from there, and he allowed me to do that. It's under his wings that I took flight. So without him, none of this would have happened. So this work, this is the starting of Murburn concept. Chlorinations by chloroploxidase occur via diffusible intermediates. That's my fundamental theory. So with that, with tribute to my great mentor, I move on. First, I move on to vision. This is a cover page credited article. So, you know the structure of the retina, right? The eye. It has multiple layers. Here's the pupil through which the light comes in. There are multiple layers of cells, and the rods and cones are placed at the far end. The rods and cones are deemed as the fundamental units of vision. Yes, all of us have studied that, right? Now, so the classical model is rhodopsin has a retinol bound to it. The cis retinol, upon being hit by a photon, it changes to transretinol. It comes out, and then there is a protein called transducin, which undergoes some conformation changes, and then signal relay is conveyed via ion fluxes, sodium, potassium, channels open, close, activation potential, all that you might have studied. So this is the classical mechanism. So this is the rod cell, this is the retinal pigment epithelium, and there are the rod cell discs. These are membrane filled stacks. So this is the mechanism, basically, as you see in textbooks as of now. It's the basic mechanisms of how we see. So the question that anybody could have asked before I asked. The retina, the rods and cones are here. Sorry, my bad. The rods and cones are there. So there are at least 10 layers of, thank you so much, my boy. The rods and cones are here. There are at least 10 layers of cells. Now, you imagine a photon of 400 nanometers from where the visible range starts. Now, this is approximately 400 microns thick. It is 1,000 times the wavelength of that photon. Now, to reach the rods and cones, by the time it reaches there, it would undergo all kinds of process like reflection, diffraction, refraction, all. So how can this be the fundamental place where your eyesight signal decoding occurs? It cannot be. This is a fundamental question. So this is what I said in my inaugural speech. You have to be critically minded for you to discover something different. You don't need to be a topper in your class. All that you need to do is pay some attention to what is going on. Get interested in something. All right? So now, let us see the structure of the proteins that are involved. This is the rhodopsin, this is the transducin. It is a G protein coupled receptor. If you know some biochemistry, it's a very, very important protein. It's a target of multiple pharmaceutical companies. Why don't you come here and sit? So that, come here and sit. There's no point in sitting there. Sit there. So, now, You see, the classical theory holds that the transducin, the alpha module of transducin, it undergoes a conformational change. When the light hits and the retinal comes out of the rhodopsin, it goes through a transformation change. These are the GDP bound and GTP, guanosine diphosphate, guanosine triphosphate. In the classical mechanism, GDP is exchanged for GTP activating transducin, and that leads to a chemical signal, electrochemical signal. Now, there is hardly any structural difference that is known. How can you explain the outcome? That's another problem. And then there is this beta transducin module which doesn't have any role whatsoever in their mechanism. So it's highly unlikely that such a huge part of protein, which is the transducing element of the photosignal, it is highly unlikely that let's do that later, please. So transducing element will have elements which are not accounted for. So what is the Murburn model that I have proposed? The Murburn model is, these are the basic elements. This is the at rest, rhodopsin, transducin, and there is a phosphodiesterase. Now these are supposed to come together. First thing is, I claim that oxygen is obligatorily involved in this mechanism. Till now, the mechanism of vision does not have any role for oxygen. That's a fundamental difference. I say that in the vision process, oxygen is obligatorily involved. Now, I will give you the evidence for that later, but I am showing you the model first. What happens is, light hits rhodopsin, retinal gives an electron to oxygen, becomes superoxide. That is directly an electrical signal. If you have a negatively charged species produced in a cell, you have a transmembrane potential difference. So that you can see as an electrical activity. Rather than a conformation change, you have a here direct generation of an electrical charge. This is a chemically tangible mechanism. All right? And then what happens is, this reactive oxygen species that is produced attacks the GDP bound to the alpha. The alpha module. Now what happens is, it gets phosphorylated. GDP gets transformed to GTP, and then what happens? GTP is taken up by the beta module. This beta module didn't have any role in the earlier mechanism. So now beta hydrolyzes it, releases GDP, which goes and activates the phosphodiesterase. This is a simple change, but a very profound change. Because this gives from light you have an electrical signal, a chemical tangible transduction. All right? So this is the difference, fundamental. So what is the evidence that I have given, my group has given? The evidence is that when you see the alpha module, the binding of GDP is at least 10 million times higher than that of GTP. So GTP cannot spontaneously exchange with GDP. GDP should get phosphorylated and then it goes out. So my theory explains that. And then the beta module of transducin, the role is, it's a GTP hydrolase, because GDP is the signal. GTP binds beautifully, it gets hydrolyzed, gets GDP and gets out of the other aperture. So all the structure activity of the protein is explained by the new mechanism. Earlier there was no explanation, concrete. So same, after the GDP is released, it comes and binds here. New sites of GDP binding are discovered in phosphodiesterase 6, and this is supported by other literature. These are all predicted. I sat there at home and predicted these things. We'll find it, you look for it, you'll find it, and we found it. That's what has happened. So that's the predictive power of the new theory. Your madam, Rose madam said, tell our researchers what they can do. So if you have good theories, you can predict and show and get credits. So now here also all this information is supported by binding energy studies. All right. So now what does this new theory allow us? First of all, when you have shortage of oxygen, you have blackouts. Your retina is innervated by lot of vascular, blood vessels on both sides, proximal as well as distal. Then why should more light give more reactive oxygen species? If reactive oxygen species and oxygen was not involved, why should light correspond to reactive oxygen species? These are experimental evidence. So then see, in our processing, you need 1/60 second. That's the processing time, dynamic processing. Whereas the classical cycle, if it has to go through the retinal pigment epithelium and all that, the classical visual cycle for which the Nobel was given, it would take hours and minutes. This is subsequent process, the reactions that I have proposed. So, the myelinated neurons, they conduct faster than the non-myelinated. How is that? The classical theory requires sodium-potassium ripple-based movement. Like that, sodium goes in, potassium comes out, and a ripple-based movement. In my theory, it is a movement of electrons along the axon. So that is a very simple theory. Here, it cannot explain that because the myelinated sheath should slow that down rather than speed it up. Here, since there is a sheathing, there's an insulation, it goes faster. Simple. So, how does the conversion of photophysical cue to electrochemical signal? I said that generation of superoxide. Then the important aspect is oligomerization. Rhodopsin, there is oligomerization. Why is oligomerization? Because if you have a radical intermediate to harness that, it's easier. When there is nearby, you have radical activity confined and the radicals interact and reactive species interact among themselves so that the retinal regeneration is more efficient. Things like that. So, it has also been seen that rhodopsin gets phosphorylated. In my theory, the superoxide can enable that. Also, resolution. If only the later part of retina is involved, you cannot explain the resolution. So in the new theory, what happens is, all these cells are involved in depth, perception, in resolution, all that. And in the new theory, this is an ultimate source of electrons. So, modern day LED screen, there's a projector of light from behind, something like that. It's a very different mechanism. So, the eye retina functions very differently. All these cells are being used to tap the signals, and this enables us to explain the structure of the eye, because you see this structure, otherwise the light comes in here and action potential has to go the other way around. It doesn't make sense. If this were true, you would have the structure like this. Rather than having multiple layers, you would have had the cones and rods facing this way directly, and you can have a diffuser if light intensity was a problem. So this theory explains the architecture of the eye, the structures of the proteins, the kinetics, all those aspects. Earlier, we couldn't address so many questions. So that's what is written here. Ganglion cells are all multiple cell layers could transduce photons. Rods and cones are ultimate sources of electron. The electrons move along this way from dendrites to synapse. All right, that's the signal. Otherwise, what happens is here earlier it was sodium-potassium movement. So that brings me to half of my talk. I started at about 10:57, right? So half my talk is done. I took a lot of time. So here now I come to what is the fundamental role of respiration. What is oxygen needed for? In this part, this is also a cover page credited article in Progress in Biophysics and Molecular Biology. The classical mechanism has the major mitochondrial proteins. How many of you have studied this? Please raise your hands if you have studied this. Electron transport chain, rotary ATP synthesis. This is all incorrect. I will tell you in the next few minutes, you can see it for yourself why it is incorrect. So this is something like this. These proteins, as electrons are going through these kinds of mega protein complexes, they pump out protons. That's the current belief. And then when the protons come back via complex 5, ATP is made. Right? Now this model is something like a dam. The water comes in, you have a turbine, and then the water is once again pumped out. It's the model like that. And for this, you need to have the proteins arranged along the crystal like that, as people have said. Now, this is an introduction part. Now the first thing that even a 12th class student can understand. Very simple. Mitochondria is a micro-dimensioned organelle. So you can use Avogadro's number. This is what I said when I talked about in my introduction. There's a girl sitting there. She is looking at her phone perhaps. Hello? Here. Yeah. I know now why you are sitting at the back. So don't do that. If you sit at the back, don't indulge. You are losing an opportunity. This is something even a 12th class student could have questioned. This is for which Peter Mitchell got his Nobel. You get interested in what happens around, you might have a chance to do something significant, important on your own. So, if you see, a mitochondrion has finger countable protons. It's got such, because water does not dissociate completely to protons and hydroxide ions. It's got a very low dissociation constant. So at pH 7, pH 8, the concentration of hydrogen is proton is so less, if you calculate, it comes to, in cyanobacteria or in mitochondria, it comes to less than 1 or less than 10. Right? So, but then these proteins that you saw, they are in tens of thousands or lakhs. So can tens of thousands of lakhs of proteins be involved for pumping protons? The obvious question is no. So, the chemiosmosis, rotary ATP synthesis, 2 Nobels. Irrelevant. What is this important for? This explains how you produce ATP in your cells. It's a fundamental powering logic of each and every single living being on this planet. It's that important. If you are a biologist, there is no way that you cannot be interested in this. Next, chemico-physical viability. Next is, now the classical theory has complex 1, complex 4, pumping protons using the NADH plus oxygen reaction. This is an exergonic reaction. Now when it comes back, it is supposed to make ATP. And it is also supposed to generate heat. How come? Thermodynamic, there is no sense. Suppose you have a pressurized balloon. You are pumping out something from that. It cannot come back into that same thing and do work. It's very difficult. These are the ideas that were sold. Alright? Now, if you see in terms of electrochemistry, if you try to maintain a positive and negative charge across a membrane, the movement of the ionic species have to be limited in dynamic state. You cannot have here protons are going out, coming back, electrons go out, come back. It cannot work like that. That's not how a question in electrochemical system works in steady state in reality. This is all a story that has been sold. So this story can be questioned. Even the calculation can be done in a reverse way. Reverse way in the sense wherein you calculate from the voltage at which mitochondria works. You see that when you do that, even then the calculation does not stand. the working of the proton-based rotary activity, it has been compared to a rotor. Do you think a molecular rotor can self-assemble? Very difficult. Try to assemble a rotary thing in your own lab. Motor, try to assemble that. How many unidirectional operations you have to do? And here it says that when protons come in, it makes ATP. When protons goes out, it is hydrolyzing ATP. Now, it is like imagining this. Now you have a juicer. You try to put a fruit from the top. You get pulp and juice from the bottom. Can you change the configuration in some way and get the fruit formation from fruit and pulp? That is what is advocated here. It was advocated, it was accepted. It's thermodynamically infeasible. The fruit gets made, but it's made in a different way, not in the exact opposite way. Now the proton comes inward. What is it stopping it from going outward? Right? These are the kind of questions critical-minded people ask. So this does not make sense. So what is the Murburn model of respiration, the new model that is published? It says that complex 1 to 4 are ADP binders, and they are ROS generators, reactive oxygen species generators and utilizers. ROS are considered as bad things in drug metabolism, in cancer, in immunology, everything. They are considered the villain, reactive oxygen species. Why? Because they lead to aging, apoptosis, so many things. Of course, we'll come to that later. But then, this is, what happens is, ROS is produced, so ADP is attacked by radicals, and then you have ATP formation. ADP and phosphate get attacked, and then you have ATP formation. It's a very simple bimolecular chemical reaction. There is no overt intelligence, there is no extra gambit kind of a thing. You don't have to pump out anything and then they have to come back, nothing like that. It's a very simple reaction. It's something like a cooking setup, a simple hearth. You have heat, flame, you cook, and the transmembrane potential difference that you, in the earlier model, transmembrane potential was supposed to mediate ATP synthesis. How can a potential difference mediate ATP synthesis? Potential difference is a global phase phenomena. This is a ATP synthesis, is a localized thing happening at the enzyme, purported enzyme. So this is a simple model. Now how do you explain the protein structures? You see, this is complex 3, this is complex 4, this is complex 1. In the classical model, there is no role at all for this. So the structure should have been like that. Compound 1, it need not have such huge matrix-word protruding arms which can produce reactive oxygen species. Why such complex structures? These structures are not explained by the classical model, but they are explained by the Murburn model, because these have ADP binding sites and reactive oxygen species are produced. And see, now you relate to the fundamental mechanistic model I said. Here, a diffusible reactive species is generating the reaction outcome. Did you understand that? Now, the structure of complex 1. Complex 1 structure shows that there are openings, pores, ADP binding sites, and the classical membrane, proton pump theory says as electrons are passed here, protons are pumped here. There is absolutely no connection whatsoever. This is a microsecond phenomena, this is a millisecond phenomena. And the overall reaction in the classical theory, it's thermodynamically non-viable. Delta G is positive. My theory, it's negative. And this protein can function independently. It does not pump out protons. There is no need for protons in this way in cristae, in the mitochondria. That's a fundamental argument. Same complex 3, I told you. These have pores directly accessing. Now, if you believe in the classical theory, there is something called Q cycle, complex cycles. And for a very simple outcome, you need multiple dozens of steps. All these are currently taught. All these reactions are thermodynamically non-viable. They are non-viable. Here you have viable reaction, very simple. It explains the structure and the reaction chemistry and thermodynamics. Sorry, my bad. Yeah. The complex 5 structure, the Murburn model says that it is a chemostat. It uses ATP to bring in protons. It's very simply supported. Boyer only found one ADP site. The Murburn multiple adenosine binding sites, and this way it can help ATP synthesis also. Partially reacted can sit here, and when the protons come, they can make ATP. So it uses ATP to open the pore to let proton in. NADH has one proton but two electrons. So proton is deficient. The deficiency is met from outside. So this is serving as a dynamic chemostat. That's the role. So in the new theory, complex 5 is not directly needed. It is a protein that evolved at a later stage. So this is a very simpler, much simpler system. And then complexing, like what we saw in rhodopsin, the same complexing is seen in multiple proteins here. That is explained by Ross dynamics, reactive oxygen species. So now you have Ross produced here. If all these things get together, Ross are released, they can help each other's activity. That's the explanation. Most importantly, oxygen's reactivity and mobility. Now, here the classical theory has one protein, one redox center. It moves one way, in particular way, chartered way. How can it move? Oxygen is present everywhere. Oxygen's mobility is 10 to the power 3 angstrom per microsecond. Now, I told you in my inaugural talk, you need to be watching out for such physical feel. You know 4 elephants cannot go into a car even though there are 4 seats. Similarly, if 4 electrons go, they cannot go like that. If you remember, it cannot go in an ordered way. There is no arrangement there. And oxygen is there to shunt all mechanism like that. So, there are 31 electron agents in the respiratory chain. Why should that 1 electron chain be there when the structures don't agree with it? And now you see cardiolipin, you can explain. The cardiolipin has high amount of negative charges on the surface. It is to restrict the mobility of superoxide. Oxygen gets reduced, it makes superoxide. So, and then coenzyme Q. If coenzyme Q was a mobile relay of electrons, here, coenzyme Q is supposed to relay electrons like this. It's supposed to go as though it has intelligence, like that. If in that case, it should be small. But the coenzyme Q that is found preponderant in the membrane is coenzyme Q10. Large, very large molecule. So, in the Murburn model, this is used to pack the membrane for electron sponges. Cytochrome C is an electron sponge and coenzyme Q is an electron sponge. Most conclusive evidence is, I showed through my research collaborators, that you take ADP and phosphate in a test tube, you add superoxide, reactive oxygen species, you get ATP. Simple reaction. You don't need anything else. Conclusive demonstration of a simple theory. ATP formation is a very simple reaction. It's not a mechano-electrical stunt as currently thought. Now, before me, Kathleen Mailer, a woman scientist, she had shown that when you take mitochondria and you subject it to some reactive oxygen species generated from xanthine oxidase, you get ATP also. And this process is inhibited by superoxide dismutase. So, a reactive oxygen species eating enzyme inhibits the ATP production. So, clearly, in vitro, in vivo, both clear. And it is known that the more NADH and O2 you give to mitochondria, you get more amount of reactive oxygen species. Increase in ROS correlates with TMP. Now, if you are driving a scooter, when it generates sound or smoke, you don't say that the smoke or The sound is what drives the engine, right? Currently, what they believe is like that. They see that the transmembrane potential is what drives the ATP synthesis. You saw in the rhodopsin, superoxide is produced, the negative thing, that gives up membrane potential difference. Here also, the transmembrane potential difference changes with respect to increase in ROS, and the more ROS, you get more ATP. No ROS, no ATP. Conclusive. There is no other argument about this. And this overall equation can afford the explanation how one molecule of NADH can give 3.5 molecules of ATP, whereas the classical theory cannot. So these are conclusive. And then heat, not just energy. If you don't have 37 degrees centigrade, your reactions don't happen. How is the heat produced? So the classical theory requires protons, fatty acids, and the dissipation of transmembrane potential. This is a very simple theory. Uncoupling protein has loops like this, which are rich in arginine and lysine, positively charged amino acids. They can help the transport of negatively charged ROS, which can meet protons from there, making water. This is a highly exothermic reaction. It's a simple chemical explanation that agrees with the structure of the uncoupling protein. So this is the one that takes the cake. Cyanide. You all biochemists, they confirm mechanism using inhibitors, right? Now you know cyanide kills. It inhibits a vast majority of biological activities. Now it does so at milligram per kg or micromolar levels. Now, if the classical mechanism says that cyanide binds to ion centers, preventing the utilization of oxygen, right? If that is so, you need enough cyanide to bind. So now suppose I have 100 heme centers in my body and I take one cyanide, I still have 99 heme centers, right? Why should it inhibit? This is the reality. You have to, for binding based effect, you need gram per kg. You need to eat cyanide like this for it to be effective. But that is not needed. And the effect is very quick. So the Murburn theory says that the reactive oxygen species that is produced is the one that makes ATP. And what cyanide does is it reacts with the reactive oxygen species, preventing the usage of the ROS to make ATP. So you don't need food for 2 days, you don't need water for 2 days. Oxygen is not needed to make water. The classical theory says that oxygen is needed to make water. Imagine how much impact, how many people's lives we could have saved if people knew why oxygen is taken in COVID. When they administered, they found, the practitioners found that they were doing more harm because they don't know the biochemistry behind it. It is not oxygen that is needed. It is the reactive oxygen species that are generated from oxygen, and that is a very critical, crucial equilibrium-based effect. So this is the reaction. So you see how facile this reaction is. Minus 275 kilojoules per mole. Now, if you need to understand that, you need to understand all these concepts. As biotechnologists, if you just say, I cut one gene with this restriction enzyme, that, you will not be able to understand this. So this is what I said in my inaugural speech. You need to be aware of the units. You need to be aware of the conceptual understanding. Now, very clearly, the cyanide explanation for Murburn concept is supported by experimental data. Carbon monoxide is a much stronger binding agent of heme than cyanide. The data is there. But carbon monoxide, even smokers have 10% in their blood. 40% doesn't even take your senses off. Whereas 2 to 3% cyanide, you're dead. Not just this, even in photosynthesis, madam. Cyanide inhibits photosynthesis. In the photosynthesis also, the mechanism is based on reactive species. That's the reason. How cyanide inhibits a wide bevy of physiologies. So, and too slow. The cyanide binding, if you see, the K on, the values, they are very slow. You need minutes and hours for effects. Whereas within a few seconds, done. Life gets done. That's because the radical reactions are much faster. They are bimolecular. That's the reason. So it's not just I who doubted. There were cynics before me. I pay tribute. It's a pity that you people have to study these kinds of theories. Research is going on still using these kinds of ideas. So I pay tribute to the people who doubted. Can you see something interesting about these papers? They are all single author papers. A consensus can be generated about people. They can believe, but then discerning scientists think in their own shoes and think for themselves. That's what is shown here. So now I have shown you examples wherein that Murburn concept was applied. I would like to tell you a few more words about Murburn concept. Here is the classical enzyme mechanism. You have the Fisher's lock and key, the Koshland's induced fit, wherein the substrate comes and binds at the active site. S stands for substrate, it binds, that is the active site. The inhibitor can also come and bind at the active site. So active site, inhibitor inhibition. There is an allosteric inhibition also. So this mechanism gives you high substrate selectivity, specific redox partnering, and integral stoichiometry. What is integral stoichiometry? When you study a reaction, 2A reacts with 1B gives 1C plus 2D, something like that. That's an integral stoichiometry. But then when you do many of the reactions, you don't get integral stoichiometry. You get 0.5, 0.72, something like that. You get, those things are explained by the Murburn concept. And how can an enzyme interact with diverse kinds of substrate? When the recognition is not through active site, but if it involves a diffusible reactive species, you can get all these outcomes also. If this reactive oxygen species or diffusible reactive species is consumed by the intermediary agents in the milieu, you will get. So this is a stochastic scheme. Stochastic means chance-based, probability-based. The earlier theory is deterministic. I am saying this is also true, but this is a larger scheme of events. Do you understand? I am not negating this. I am just saying this is a larger scheme. This also happens, but this also happens. Now, what does it translate to in terms of your mechanism? You might have seen Michaelis-Menten kinetics, the hyperbola with respect to substrate, or even the time course is usually like this. Now, you might get some reaction profiles like this. Classical enzyme kinetics mechanism cannot explain this. The Murburn model explains it. And then there is, this is at the level of metabolism. What does it translate to in terms of physiology? You might be given a drug, so let's say 1 milligram drug. It produces great effect in you. But then somebody administers you 10 milligram or 100 milligram, nothing happens. How can a small dose give a greater effect than a larger dose? There is no explanation for that in conventional biochemistry, because both ligand receptor binding and enzyme substrate catalysis, they are basically that kind of profile. Right? If there is an inhibition, you might get like this, but you cannot explain haphazard. If you have a diffusible reactive species, it's a radical, it gets stabilized at discrete concentration ranges due to the equilibrium involved. So you can explain. Very simple explanations. So idiosyncrasies, do you know what idiosyncrasies is? It might affect Rajgopal, but it might not affect Afzal. Something like that. How can you explain such differences? Hormesis is the effect of low dose effectiveness over high dose. Now, coming to the final few slides of my talk, please pay attention. This is a fundamental concept that I want to impart. So, our cells, our body, all living beings that you see, they are made of cells. Cells fundamentally function as per the new theory there are fundamentally four forces. What are the four forces? Gravitational force, smaller, longer range nuclear forces, and electromagnetic force. Electromagnetic is the force that life uses. Now, in that, we need to be in a position to take fuel, just like a chemical combustion engine, and give out products in a dynamic way. Similarly, cells also do that. I say they do mute burning. In textbooks in 8th, 9th, what is the difference between combustion and respiration? If you remember, you have studied this. Practically, there is no difference. At the school level, it has to change. So now it's an oxygen based, just like burning, but it's a controlled burning. That's all. So the control, earlier reactions could get controlled at the enzyme active site. Here the reaction control is based on many other aspects. Delta G of formation, thermodynamic pull, dynamic partitioning, dielectrics, proton, so many other physical chemistry based rationale. Right? So now what has this concept given us? It has given us new and more sensible explanation in enzymology, biological electron transfers, dose responses, drug metabolism. All the drugs that you take, it is $1 trillion industry. Our liver metabolizes that. The reddish brown color is because of cytochrome P450. If you eat lipid, proteins, carbohydrates, they are metabolized by the carbohydrates, lipases, and proteases. They have evolutionary memory. You are taking a molecule like ritonavir, an antiviral drug that we made. How does an enzyme recognize that? There is no real recognition. It is by this kind of a mechanism, diffusible reactive oxygen species, which cytochrome P450 use. So, multiple way. How can hemoglobin, which does not, erythrocyte, it does not have mitochondria. How does it make ATP? Hemoglobin is an ATP synthase, the tetrameric structure. All that I have explained. How does LDH function? LDH, you have in muscle 4 folds higher LDH than in liver. Then why is the lactate formed in the muscles transported to the liver? Why? As per the classical mechanism, LDH should work reversibly. It doesn't work. That's why it has to get uricycle. You remember that? Cori cycle, sorry. Cori cycle got a Nobel, they got a Nobel, husband, wife, they got a Nobel in 1940s for that. So all these absolutely new explanations. I'm coming to the end. So basically, Murburn concept is a ubiquitous principle. It is found in all parts of cells, working everywhere, in diverse membrane systems, and now the perception has changed. You need not just restrict oxygen's role in active site. It's in Murzone. Reactive oxygen species are not merely toxic waste, but they are essential intermediates. Then substrate and additives are only working at active sites or allosteric sites. Now we have to think about multiple interactive equilibriums. So molecular interactions are always affinity-driven complexations in the classical biology. In the Murburn paradigm, bimolecular collisions with or without, like that. So in the classical system, you have to think of a product being formed in only one way. Here it is in multiple way. So these aspects have to be considered. Now I'll tell you something. It is very fundamental, very important. Diffusible reactive species are the elixir of life. If you see a knife in a kitchen, there are places to keep the knife, okay, racks. There are gloves to handle. There are cutting boards. Do you infer that knives are bad, knives are dangerous, we cannot use it? Knives are dangerous, but the cutting board, the gloves, the racks are there to use it in a wise, in a systematic, in a controlled manner. Similarly, the presence of catalase, peroxidase, superoxide dismutase, all that in soluble milieu doesn't mean that reactive oxygen species are purely bad. Every molecule has contextual relevance. Amidamayal, amradam visham, we say, no? Same thing. So, how is this burning then otherwise affected? When you take a cloth, daub it in oil, set it afire. The cloth burns only after the oil burns. So you can still have control. Now, but if it burns like that, we'll also get burnt over a period of time, which is why we have to die, which is why we age and we die. It's an inevitable consequence of the operational relevance of Murburn concept. Okay, so these are the reactive oxygen species. Now, this new theory appeals to energetics, kinetics, mechanistics, structure, distribution, evolution, probability, Occam's razor, all the criteria of science, but it does not appeal to aesthetics. Okay, now I'm coming to the end. The end is, I want you to see this. It's a very simple poster. All living beings are made of cells. Cells use respiration or photosynthesis for powering. Now, of the four fundamental forces, we need to use electromagnetic force, and the cell has to work as a coherent unit. The classical theories require nucleus for regulation of units, and it requires proton pump. Cyanobacteria are so small they do not have even one free proton in their cells. And then RBCs do not have mitochondria and nucleus to control them. But RBCs are relevant for 120 days. So how do they work? We need a completely new theory. There are no protons. So that new theory is Murburn concept. Murburn concept is a fundamental pillar of life. You know the cell or protoplasm theory. Cell is the fundamental unit of life. 1. 2 is the central dogma. Genes make proteins. Proteins and genes control cellular activities. That's the second one. And the third one is Murburn concept. Murburn concept is the one that allowed the evolution, origin, sustenance, and is also responsible for the termination of life. It is that part of chaos that is harnessed by cells. There is a lot of disorder, but even the disorder, if you study chaos, even see a fern's leaf, has anybody studied fractals? If you know, fractals are resulting out of chaos theory, right? But even in the fractal images, you will see order. Similarly, life's order uses the chaos. So basically, cellular molecules and ions are in dynamic equilibrium, and they use diffusible reactive species, and then oxygen serves to generate these diffusible reactive species by the principle of effective charge separation. You need charge separation because that is the way in which electromagnetic force can be harnessed by the cells. So now oxygen-based theory, why do we have oxygen-based life? Because carbon dioxide, it can go out quickly, like the vehicle lets go of the gas, no? It can go off. And then the water is the other product. It can equilibrate. So the water can be used to wash away the products. So imagine a condition when there is not much specificity. Life has to evolve before gene. This is the fundamental principle that allowed some selectivity before the selective mechanisms came in. So DRS enables cellular powering and coherence. How they connect the different parts of the cell. Okay? So metabolic and physiological functions. So the last slide, I've ended my time, 1 hour. I would like to thank you. Before I thank, I want to urge you, each and every one of you here, please, my dear students, see life on an open, optimistic, white, plain canvas paper. You can write your own history. I am from an ordinary background from Kerala. If I can challenge so many concepts, so can you. Tatame pucha pucha. Parroting is not what a scientist does. You need a question. When I was studying like you, I questioned certain things. And then I took it upon myself. So that exploration aspect is very essential. I thank my students, Surjit, Daniel, Abhinav, Afsal is here, and Vivian, my collaborating students, and the international collaborators. I have Nikolai Barin of Russian Academy of Sciences, Lauren Jakin of Karel de Grote Hoge School in Belgium, Yan Yovu of Chinese Academy of Sciences, Hirohisa Tamagawa of Gifu University, Japan. Thank you so much for your patience. Thank you so much for your kind attention. Thank you for bearing with my requirements, moving ahead when I wanted to have you move. Thank you so much. Thank you so much. Thank you so much. Thank you. I am open for questions.
Now invite Dr. Kavita AO, Controller of Examination and Assistant Professor of Department of Biotechnology, to give the token of appreciation. I also invite Professor Dr. Kelath Murali Manoj to receive the token of appreciation.
Membrane potential. That's what this shows. That's what this shows. This is conclusive demonstration. I forgot about it. So